the business development associate here, and today we are glad to have Matt Newman, who is our VP of Business Development, to start today's webinar. And I think I will turn to the presenter for a 30-minute presentation, and after that we will have a Q&A session. So if you post any questions during the webinar in this session, our presenter will pick up a few. And if your question is not being addressed during the webinar, we will definitely get back to you through emails in the following couple of days. So now we will get started, and I will turn to our presenter here. Matt, welcome.
Okay, thanks, Vivian. As Vivian said, we're going to post the slides and a video of this webinar later on. Somebody already asked me if we could put the slides in the GoToMeeting handouts, and normally I try to do that, and it looks like it's not going to let me do it since we've already started the meeting. So we'll send out the information after the meeting as to the slides. So the topic of today will be mostly data, almost primarily data. We have new data for both ImmunoLand and CVMLand. And then I thought I would just show you how to use the data in different ways. There are some users that have purchased ImmunoLand, CVMLand, and OncoLand. So we'll talk about creating a virtual land of all three and some of the queries you can do across different disease areas as well. So getting right into it. In ImmunoLand, there are nearly 650 new RNA-Seq samples in the land. About 8,000 new array-based samples, and we've processed 130 or so new geo projects, and that's across 401 new comparisons. As a reminder, a comparison would be for each individual data set. You're going to have multiple comparisons potentially comparing different groups, so treated versus control, disease versus normal, responder versus non-responder, that type of thing. Everything's been fully curated as before. And is now available, or will be available when your system's been updated. As usual, I'll switch back and forth between slides and the PowerPoint, and so I'll just show you some basic stuff that's going on in ImmunoLand now, and then switch back and forth. So this is your updated ImmunoLand sample distribution. You can see all the different disease categories. Obviously, you can change this to any type of grouping. So if you're curious as to the individual diseases, for instance, you can do that. It kind of gets a little messier when you do that. So we usually use disease category. For those of you that are interested, disease category is just a higher level disease state and kind of cleans up the data when you're looking at it. I thought that I would show a view that I don't normally talk about, just because it's kind of useful for understanding what's actually inside of each land. There's a view called data availability, and if you click that, it'll use your grouping and it'll show you how many samples belong to each of the different data types. And for something like ImmunoLand, you essentially have two data types. You have expression data, and then you have the RNA-Seq data. RNA-Seq itself has 6 different data types. But in almost all cases, these numbers should match, with the exception of paired infusion, where there's going to be less samples usually, because not all RNA-Seq samples are paired. But this is kind of a useful, table here, just because again you can switch this between different things. So if you're just curious, how many RNA-Seq samples are there in, asthma? You can very quickly answer the question that there's 29 total RNA-Seq samples in asthma, and so forth. If you wanted to know, what types of tissues do we have in the land, just type in tissue, and then we can get a good look at the total number of tissues or samples per tissue. So again, if you were looking for RNA-Seq, for instance, you might filter, or I'm sorry, sort, and so you would see, the majority of our RNA-Seq samples are related to blood, and then you've got mucosa, skin, ileum, lung, for instance, and then that'll be similar but not exactly the same for for the expression data as well, the microarray data. So just something to keep in mind, as I know that there's a lot of times there's questions about what's actually in the land. There are ways to explore this that I probably haven't featured as much in the past. So moving right along, if you want to see what comparisons are in the land, we now have about 1,600 comparisons, and there's a view for this that I've covered in the past. This basically just shows you by default, by tissue, how many comparisons there are, and then we're coloring by the category, and I always mention the category, but it's pretty important. You know, how many comparisons are there for disease versus normal, for responder versus non-responder, treated versus control, and you can see that, you know, in general, probably disease versus normal and treated versus control are the big categories. This one doesn't respond to grouping, but you can always change the columns that you're using here. So if instead of tissue you wanted to use, you know, disease category to get an idea of where things are, you could do that as well, and you can see where the comparisons are. And again, you know, relate that to the legend. So these are just, I know I've covered some of these things in the past, but you know, as we continue to update the data, as there's more data, it becomes harder to understand what's in the lands, and these views become more useful. Should also point out that there is, when you're in the comparison setup, there is a button that says View Filtered Table, and this will show you all of the projects, so the geo projects and sequence read archive projects that are actually in the land. So if you just wanted to get either a list of all projects, or if you filtered them for a particular disease, you can do that straight from here, which is also useful. And of course, you always have that ability in the sample world as well. When you're there's actually a sample details view that'll show you everything. There was a question that came in: are all the array-based and RNA-Seq-based experiments all need to be using the same genome reference? Yes, per land, everything goes into the same reference library. And the same gene model as well. Okay. Oops. There we go. Okay, so this is I've done searches before. My favorite gene to search is SERPINB7, so we'll just do a search. And I just wanted to show that we have even more data than we had before. And so you'll see a lot of the same queries, and again, I don't think framework has changed much since the last time, but you can identify, you know, interesting experiments this way. This is an arthritis experiment, and you can see it's comparing inactive JQ1 juvenile idiopathic arthritis versus normal control. And of course, we still have all the other comparison types as well. So this is disease versus normal. And there's also treated versus control and responder versus non-responder, and so forth. We also have a view that I don't highlight a lot called all comparisons. So it's a default view here, right here, all comparisons. And it actually tries to combine everything. So it's a lot to look at by default. But you know, if you were only interested in particular tissue types or particular disease areas, you could use these filters to filter for particular things. So, for instance, if I had, you know, certain disease categories that I wanted to focus on, for instance, IBD, other gastrointestinal diseases, and maybe, you know, arthritis, I could filter, and then you get a kind of a nice clean picture. And where it really becomes interesting are cases where you have disease versus control comparisons going in one direction and treated versus control comparisons going in the other direction. A lot of customizations and different filtering that'll be useful to you if there's particular areas that you're looking for. And you'll see that, you know, overall we're continuing to increase the amount of data in the lands. So that's ImmunoLand. I'll come back to some things later on, but that's the main portions of that. CVMLand, as you guys know, is our latest land. focused on diabetes and some other vascular and cardiovascular diseases. We have about 600 plus new RNA-Seq samples in CVMLand, which is pretty exciting. This land will only continue to grow. Still not quite as big as ImmunoLand, but it's starting to get there. 3,000 new array-based samples, almost 100 new geo projects, and 225 new comparisons. And so without covering everything that I just covered before, I will just show you this. Again, this is your sample distribution, and then you have the ability to go to the data availability and look at that, and look at how many samples there are. There's actually also a comparison availability, which will show you by disease category how many samples you have in each comparison. You know, potentially this could be expanded to other groupings, but right now it's just showing disease category. But the take-home point here, I think, is that we're increasing the total number of samples and comparisons in CVMLand, and I feel like it's becoming pretty useful. It was a little sparse to begin with, and now, you know, as we've increased the RNA-Seq especially, I think it's become more and more useful. So just to show you, you know, some of the types of things that we can do here. Again, searching a gene. Let's say we have a gene CCL2. And so we get this nice plot here. What's cool and what I haven't really illustrated before is the interactivity of the legend. So for instance, selecting white adipose tissue will actually select any points that actually are from white adipose tissue, or visceral adipose tissue will select these points automatically. And so, you know, it's kind of interactive. You can actually change the coloring as well. For those of you who have never used our variable view, there is a change symbol properties option. And so if you wanted to color by, you know, cell type instead of the tissue, you can change that. And now you'll see, you know, individual cell types where available. Actually, the better column to color by there, if you wanted to change it, would actually be sample source. So it's a combination of cell type and the tissue type. So now, if I was looking for, you know, pancreatic beta cells, I could select it, and it's going to go right to all the tests that are related to that. So I think that's kind of useful. And you'll see it turn. It's hard to tell with one dot, but it'll actually turn red as you're selecting the points. Okay. Just as a reminder, you have the ability to select and browse further each comparison. So if there's a comparison of interest, this obesity versus normal experiment, for instance, I can select it, it turns red, and then if I click Browse Selected Comparisons, it's going to take me to a visualization that shows a volcano plot for this particular comparison, so I can, you know, interactively select genes and that type of thing. But what I wanted to point out was that the enrichment analysis, the gene set analysis plot and table, can be pretty useful for taking a comparison that you find interesting, obesity versus normal, and trying to find out, is there anything else out there that has an overlap to the same genes? And I've mentioned this in the past. What this plot will do is it'll give you a score, essentially a p-value, inverse p-value. And so the highest correlations are obviously going to be the same project. It looks like we've got the exact comparison that I picked, and then there's another one that is on a different platform that shows the same overlap in genes changing. But what you'll also see is, you know, there's this one here in heart disease, acute myocardial infarction versus normal. That's showing a huge overlap, at least in upregulated genes. You can see here it shows how many there are. So this is, you know, this is a way to take one comparison in CVMLand or in ImmunoLand and really expand it to everything else. You also have the ability to incorporate your own gene sets into this as well. So I don't know if I have any here, but if you're in the main CVMLand visualization, there's an option called, yeah, I don't actually have one in here. I'll show it for ImmunoLand. There's an option called gene set analysis plot that will actually let you put, using your own gene sets, find related data sets as well. And this is an area I'll talk about later on, but we're going to try to expand so that there's really only one significant one here based on my input list. So it's not a great related, not a great input list. Let's try this one here. Yeah, this one has more significance. There's a whole lot going on here. And you can, again, select points and see the tables. There's a full table, too, that you can look at for all this. So I'm not sure how much this feature is used. It was new last quarter, but I just wanted to highlight it again because I think you know, it's going to be a way that you can take your own data. You know, there was the question about if you had to have the same reference genome and same like if you're taking your own experiments and trying to put it in, if you had to match the reference genome, this is one way to get around it. So, you know, if you had your own experiment and you want to take that list and see what's changing in the public, that list just has to be based on gene symbol. So it doesn't really matter what the reference library is for doing something like that, at least. Okay. Covering another area that we don't normally cover, and that is, I'm going to go back to CVMLand here. That's the concept of comparison sets. So I think most of you guys have seen me talk about sample sets, you know, creating subsets of samples by selecting things of interest. But what you might find when you're doing your searches is you have a bunch of comparisons that you want to then look up, like either in the analysis tab, which is what I'm going to show you, or you can actually save them for later so that you can just search for these same sets of comparisons. So if you're, you know, if you're always looking at the same set of obesity projects, you know, you instead of just doing a search and then filtering for those obesity projects each time, you can actually use the comparison set concept to have it saved in the system. So for instance, if there was a particular project, I can use the project tab here, but if there was a particular project that you're interested in, which is going to be GSE 18732, I can filter for it. I might look at my comparison tab now. So I just have one comparison, actually two comparisons. And then what I can say, so there's two here. One is glucose intolerance versus normal, and one is type 2 diabetes versus normal. And I might want to take these and create a set out of this that either I'm going to analyze in the analysis tab or I'm just going to save it for later. So there's a button in the bottom left called Create Comparison Set. You get a bunch of different options. These are selected. I very easily could choose either Create Comparison Set from selection or from filter. I'll just do selection, and we'll call this muscle glucose and diabetes 2 versus normal. And we'll tag it with some sort of tag. The tag doesn't really matter other than for organizational purposes. And then we'll say upload. And it should create a comparison set for me, I believe, that I can then use to download later. So if we look at my comparison sets here, you'll see that it created this set here that just has a list of comparisons. And then the nice thing is this becomes searchable as well, so I can type in that, and it'll take me right to it. What I wanted to demonstrate now was that you have the ability to get all of this data out without just using the Save As buttons in here. There is a download menu that is useful for downloading both the comparison data and subsets of sample data. So here we're going to download the comparison data to our local analysis. We're going to download selected comparisons, all genes, and you just have to have a project open in the analysis tab side of things. I'll grab the gene level data, the full metadata, and the original data, and then we'll load the comparison. Click download. And usually this takes a few seconds. And what you'll get is all the data in the Solution Explorer in Array Studio. And you might say, well, why do I need to do this? What will be the reason for coming over here? There are a lot of times there's more functionality in the Analysis tab than might be in the Land tab. So doing different correlation analyses, doing different manipulations to the data. So this is basically giving me everything that we looked at before. It's giving me the log2 fold change, raw p-value, adjusted p-value, and what the numerator and denominator were for the different comparisons that were done for each of the probe sets. And giving a microarray data set that actually has those fold changes in it too. And you can, if you had, say, 10 of these comparisons, you could do different visualizations and compare, for particular genes or sets of genes, or look at heat maps. But again, it's things that you might not necessarily be able to do easily in the Land. You can do here. Similarly, you have the ability to create and download sample sets as well. So I'm not going to go through this live, but the same concept. If you selected the same GSE project in the sample views, you could then create a sample set from the filter. You would then use the download selected samples across all module. Grab your expression data or your RNA-Seq data that you're trying to get to, and then it puts that into the Solution Explorer where you'll get all of the data that you can work with. Again, I find this very useful for doing more advanced clustering, trying different analyses. If it's something where we did a two-way ANOVA and you thought that we should be including something else in the model, you can easily get to the and have it put in the Solution Explorer where you can do it. You can do analysis, and then of course the microarray menu, which has a ton of different modules, QC modules and analysis modules, as well as the NGS menu for some of the RNA-Seq data, becomes available, and you can work with that. There's a concept called virtual lands in our system, and it's combinations of lands. If you've purchased all three of our disease lands, ImmunoLand, CVMLand, and OncoLand, we can help you create a virtual land that actually combines everything. I rarely talk about the mouse, but we do have we have an equivalent land for ImmunoLand and CVMLands for mouse, and then you can make these virtual lands customizable. In terms of what like I'm going to show you one where I just combine these three, but there are other oncology lands out there that you might include in there. There's also the GTEx data set and the blueprint data set that might be useful for including in like a massive land that allows you to do it's going to allow you to search across all the different disease areas. So I'll show you this. You'll notice there's a virtual land tab over here that you guys probably have different stuff. I've created one called DiseaseLand Human. So essentially, it's given me, I've combined CVMLand, ImmunoLand, and the OncoGeoLand that I talked about yesterday, which is a collection of comparison data from oncology. And so you look at the comparison availability, you'll see all the different disease areas that we're searching across now. And so I could do the same search that I normally do. Let's say B7. And now we're returning it across all the different disease categories. One useful customization that I found, if you switch to the task tab here and say specify multiple profile columns, you can actually add the source land as part of your categories here, and then you'll be able to see, okay, this is interesting. You know, in immune diseases, this is definitely going up in disease versus normal. In cardiovascular diseases, this gene really isn't changing much at all, at least not significantly, and not with huge fold changes. But in oncology, we're seeing a downregulation of the same gene, and then obviously, you know, selecting data points will show you more information about those different points. So I think it's, you know, it's a nice, like if you're trying to search across disease areas, creating a virtual land like this is definitely very useful. Just checking for questions. Okay. And then, you know, associated with that, I don't cover it much, but we do have all of these underlying data views. So we have the expression views and then the RNA-Seq views. These are available for the virtual land as well. So if you wanted to just see, you know, what's the expression across all of my data sets for SERP and B7, you can get a plot, especially with RNA-Seq, where it's all relatively comparable. You can get a plot like this. And again, if you add in something like, you know, the source, and then maybe put in, you know, tissue, you can get some pretty interesting plots. And so, you know, you can very clearly see what diseases this gene is expressed in. And this is where I was saying, you know, if the GTEx data was included, you could compare, you know, expression in oncology tissues to immunology tissues to cardiovascular tissues, and then a whole set of normal tissues that you might filter for. So I think there's some really unique ways you can manipulate this data. So that brings us to about a half hour. The same searches can be done via mouse. Right now, mouse is just actually just CVM and ImmunoLand. Not there's no oncology data for mouse yet. So in terms of what's coming up, want to once again remind people that there is going to be a user group meeting May 3rd to 4th in Cambridge, Mass. We've had a pretty good response rate. I think we have over 50 attendees so far. It's only been a week or two since we announced it. It's free registration. For those of you not from Boston, we have hotel accommodations set up that you have to pay for, obviously, but we have links and there's discounts on the rooms and everything. So if you're a regular user, I advise you to try to make it to the meeting. I think it's going to be a really useful meeting for everybody to understand what Omicsoft's doing and excuse me, and also understand what like oh, excuse me. I think one of the main advantages of the user group meeting is going to be talking to other people and understanding what they're doing. As far as what's coming in the next release, that will be announced at the May 3rd, 4th meeting. We're going to be adding more data, obviously. 2 new lands that will be, I believe, in ImmunoLand. One is EncodeLand, which will focus on the ENCODE data sets. And one is a single-cell land, which is going to be focusing on single-cell experiments. So I think both of those should be pretty interesting. We'll also be doing have some framework improvements for the gene set analysis so that you can include pathways and a more general way to manage the gene sets that are actually used. So you can just have, you know, straight lists that you might stick into there and be able to compare. And as always, send us requests if there's particular data sets of interest. So just wanted to thank everybody for coming. We'll hang out for a couple minutes while we see if there's any additional questions. And as always, you can email me after the meeting or email support@omicsoft.com after the meeting with any additional questions. Okay. I don't think there's any more questions coming in, so, oh, we got one. Are there any intra-land batch effect correlation or corrections or analysis being performed? There's not, but all the except with the exception of RNA-Seq, which I think is comparable across lands. You know, most of the visualizations and queries that I was showing you were at the full change level, and that's much more comparable than, say, looking at the array-based level. RNA-Seq, everything's been processed in the exact same manner. So, you know, while it may not be the perfect normalization, it's certainly better than comparing against, you know, like if I was to look at all the chip-based data side, like across all these different data sets, that's a lot harder. But the big example I always give when I talk about ImmunoLand especially, and then it applies here, is if you take a PCA of any array-based data and you, like if you had the GTEx data, the TCGA data, and the ICGC data, and if it was all array-based and you put that into a PCA, you'd end up with, you know, the TCGA data here, the GTEx data here, and the ICGC data here. With RNA-Seq, if everything's been processed with the same pipeline, you don't see that. You actually see the tissues. You know, it's actually going to group by tissues and disease more than anything else. And there was an additional question. What data will be included in the ENCODE-LAN? Definitely RNA-Seq data. I believe that we're going to try to include the ChIP-seq data as well, but that's all stuff that'll be announced at the user group meeting. All right. Well, if there are no more questions, or even if there is, I think we'll stop there. And like I said, you can email me with additional questions or follow-ups to anything. So thank you, everybody.
Right. Thanks, everybody. See you next time.